So in the next plant hormone in the group is the cytokinin. Now if you break this word, you can understand it's a Greek language word again. Cyto means cell and kinin or kinesis means movement. So the cell movement here actually stands for the movement which is visible in the cell division. So cytokinins is basically responsible for cell division. So cytokinin is mildly basic type of hormone which is made up of adenine or aminopurines, which we have discussed in the last class, and it promotes cell division or what you called as cytokinesis in plants along with auxins. Now primarily its function is to control cell division, and it was discovered by Miller in 1955 for the first time, and it is the first natural cytokinin was extracted in 1963 from unripe maize grains, and it was named as zeatin. From this name you can make out zea basically is a scientific name of maize plant. So the first natural cytokinin in 1963 was extracted from the grains of maize plant, and that's why it is called as zeatin. It's a 6 amino purines. Then there are 18 types of cytokinins which have been discovered so far. Still more are going on, and cytokinins are basically perform the following functions. So the functions of the cytokinins are, the easiest one is to promote cell division. So cytokinin, as the name itself suggests, is essential for cell division or cytokinesis, and it promotes cell division by controlling the activity of cyclin dependent kinase. If you remember, in cell division chapter we have discussed about cyclin dependent kinase. So it is controlled, cytokinin controls this kinase activity, and thus it controls the cell division. And it also is responsible for cell expansion in the case of leaves. So the leaves will start becoming bigger in size, or even cotyledonary leaves will become bigger in size. So this is all controlled by cytokinin. Along with that, it is also involved in organ formation. So what is organ formation? We all know what happened in the morphogenesis. Morphogenesis means the body formation or organ formation. So the relative concentration of auxin and cytokinin controls the complete formation of organs in the plant, and this concept is also used in tissue culture. So if the amount of cytokinin and auxin is almost similar, then there is no organ formation, only cell division continues. So an undifferentiated mass of cell is produced. Now whenever the amount of cytokinin increases with relation to the ratio of amount of auxin, that means more amount of cytokinin, then in that case shoot development is visible when tissue culture is performed. And whenever auxin concentration is more compared to the cytokinin, it has been found this promotes the root formation. So this concept is present naturally also in the concept of morphogenesis or organ formation in plants, but this is also used in tissue culture. Now another function of auxin is to counteract something which is called as apical dominance. We learned in the previous class regarding auxin's function of apical dominance. So this apical dominance is counteracted or is removed with the help of cytokinin. So cytokinin initiates the growth of the lateral buds despite the presence of apical bud. So even if apical bud is present and apical dominance is still there, it can be removed with the help of application of cytokinin. So it promotes the formation of new leaves, and lateral buds of cytokinin overproducing mutants grow precautiously, forming shoots that compete with the main shoot. So it has been found that if cytokinin is sprayed, the lateral buds which were dormant, they become active despite of the presence of the apical dominance. In the same manner, the next function we can learn is the sex expression. Cytokinins, just like in the case of auxin, promotes femaleness in certain plants, and it also promotes chloroplast development in plants. That means the longer the chloroplasts remain, the longer will be the life of the leaf or life of a green fruit, still ripening fruit. So the longer the fruits and leaves remain on the plant, better will be for the crop formation. And it also has a control over the opening of stomata. So opening of stomata, this concept is also removed because we are not covering the 11th chapter, that is the movement of water and all. Otherwise I would have explained to you how stomata open. So opening of stomata is also promoted by cytokinin. Now what are the uses of cytokinin? So cytokinins, the best function of cytokinin is the Richmond-Lang effect. Richmond-Lang effect is the delay in the senescence. I have explained senescence before to you, that the aging of a structure is supposed to be called as senescence. So if there is a delay in the aging, then there is going to be increase in the nutrient and the sink activity. This will cause increased shelf life. So that means that the shelf life of fruits and vegetables can be prolonged. So if somebody is selling fruits and vegetables, and there are some vegetables and fruits, they ripe very fast and they have to be sold very quickly, otherwise they will be rotten. So such type of vegetables and fruits can be made to remain fresh by application of gibberellic acid, with the application of cytokinin, and this effect is called as Richmond-Lang effect. In the same manner, it helps in breaking dormancy, like in the case of gibberellic acid. It induces flowering in some plants, for example lemna as well as wolffia, and it has been found that transport, that means the transport through phloem is also promoted because of the presence of the cytokinin. So tissue culture, senescence, shelf life, all these things are controlled by cytokinin.